All right, it's 12 o'clock. I want to welcome everyone to this week's Iowa Learning Farm Conservation webinar. I am Matt Helmers, faculty member here at Iowa State University, director of the Iowa Nutrient Research Center, and faculty affiliate with the Iowa Learning Farms, which is most important for this webinar. So thank you all for taking time out of your schedule to attend, and I think we have a great webinar today. A little biased because Dr. Laura Alt is a graduate alumnus of our ABE department here at Iowa State University. She's a postdoctoral researcher, and she's going to talk to us about from rump to runoff, the transport of antimicrobial resistance in agricultural ecosystems. I think this is a really important topic and timely topic because there are increasingly questions related to this. So as always, if you have questions, feel free to type those in the chat box and we'll get those to Dr. Alt at the end of her talk. And so with that, I'm going to turn it over to Laura. The virtual floor is all yours.
Awesome, thank you. Quick shout out to all of you for taking the time to listen to this talk today. Won't spend too much time on the title since Matt just introduced me. But to quickly just kind of outline for you how this talk is going to go, we're going to begin with an introduction in which hopefully I'll answer the questions, what is antimicrobial resistance, or AMR, since antimicrobial resistance is kind of a mouthful? Why should we all care about it? And then what is the relationship between AMR and agriculture within the scope of my research? I'm then going to move into covering some of the work from my time as a student at ISU, which involved looking into using prairie strips as a management practice to limit the transport of AMR contaminants within agricultural systems. And here we'll talk about kind of the common contaminants associated with AMR. And then I'll also get into discussing 3 different research projects that I was a part of. And then finally, at the end, I'm going to try to bring everything together and kind of give you some of the major implications from this research before getting to questions at the end. So if this goes to the next slide, yep. So antimicrobial resistance, or AMR, can be defined as the ability of microorganisms, so things like bacteria, fungi, viruses, and parasites, to survive and grow in the presence of an antimicrobial drug to which they were once sensitive or susceptible. So overall, it essentially means that a drug that might have killed them before is no longer killing them off now. And in the context of the research that I'm presenting, we're very much focused in on bacteria, which are commonly treated with antibiotics. And so sometimes you may hear myself or others use the phrase antibiotic resistance, almost a little bit interchangeably with antimicrobial resistance. But just to be clear on terminology, antimicrobial resistance is kind of the umbrella term for resistance in all microorganisms, while antibiotic resistance is specific to bacteria. Okay, so why is antimicrobial or antibiotic resistance important? And why should we all care about it? The emergence, evolution, and spread of these resistant pathogens represents one of the world's most urgent public health crises. In our context, as bacteria develop resistance to our currently available antibiotics, we lose our ability to effectively treat any resulting infections. And so here I've pulled just a few recent headlines that reference specifically antibiotic resistance to try and show you kind of the relevance of the topic. But honestly, I think one of the best ways to maybe put into context some of this health risk is just to compare it to the COVID-19 pandemic. So while COVID is a virus, so not something that we treat with antibiotics the way we do bacteria, at its basis, it was a microbe that we discovered a while ago, studied for some time, but upon its mutation and with its spread into people, it became just incredibly infectious and deadly at a large scale. And I think we can all agree that when infectious microbes evolve and essentially are left to run amok because we don't have effective treatments for them, the results can be really, really devastating. And so in the context of just raw numbers, in 2019, it was estimated that antibiotic resistance was associated with nearly 5 million deaths worldwide. And again, if I'm using COVID as a measure for reference, the latest numbers of COVID-19 deaths reported to the World Health Organization are now exceeding 3.3 million. So we can see that on just like a purely numbers basis, antibiotic resistance is already causing global deaths at the same or a potentially even bigger scale than COVID. Okay, so I just took you through a bunch of information, essentially letting you know that antibiotic resistance is this big public health crisis and that it's really impacting human health. And I've essentially put all that effort into driving home this human health component. But in reality, the only way for us to really effectively combat AMR is to actually recognize it as a One Health issue, meaning that it connects the health of animals, humans, and the environment. And it's really only through kind of coordinated action from all three of these domains that we can really slow down this issue. Because ultimately, that is what we are trying to do. We're not trying to do away with AMR or antibiotic resistance altogether. What we're really trying to do is slow down its progression so that our development of new antibiotic classes can catch up. Because the thing is that our discovery of antibiotic resistance occurred really not that long after our discovery of antibiotics. I think a lot of people may actually be unaware that a lot of our modern antibiotics are products that we've harvested from bacteria in our soils. And so it's kind of logical that through proximity and exposure to these antibiotics, other bacteria in that same soil environment would have evolved mechanisms of resistance to continue to be able to survive. So what that means is that even in like pristine, untouched by human environments, antibiotic resistance can still be naturally occurring. But this is the thing, is that the more an antibiotic is used, the faster we drive antibiotic resistance. And so it becomes this kind of balancing act between managing the development of antibiotic resistance and discovering new classes of antibiotics or new treatments. And unfortunately, at the present, we've really fallen out of balance. Resistance has continued to develop, with development sometimes speeding up because of certain practices in either human and animal health. But right now, our antibiotic development pipeline is dry, and we're actually in kind of a discovery void. And so again, until antibiotic development can catch up, our course of action remains kind of slowing the development of resistance. So what does it mean to kind of have coordinated action within this One Health system? Currently, the pillars of human health and animal health really seem to dominate the focus of most AMR research. And this is likely because in both of these spheres, it can be easy to see where the progression of resistance is driven by antibiotic inputs. For example, with like antibiotic use in human medicine, in veterinary medicine, and for agricultural production purposes. It can also be easy to see how progression occurs within each sphere. So through direct contact animal to animal or direct contact human to human, especially in settings like hospitals or crowds. And then I also think it's pretty easy for people to like conceptualize the exposure and the crossover that can happen between these two spheres. So in instances of keeping pets or being involved in any sort of animal husbandry, in handling animal waste, or even just essentially the fact that we consume animal products. Then contrastingly, research involving the third pillar, so that environmental health, can often be really difficult to resolve. Because as I mentioned on the last slide, the development of AMR in the environment is actually a naturally occurring phenomenon that is really just intensified by interactions and inputs from the other two pillars. And so the work that I'm presenting today is actually going to focus in on a subset of the relationship between animal health and environmental health, and that it's kind of specific to the agricultural sector. So like I said, there's numerous pathways for AMR to develop and then spread to the environment. But the pathway that we're going to be focusing on today involves the excretion of resistance contaminants in animal manure, and then the use of that manure as a fertilizer. And so just to quickly walk you through what this looks like, antimicrobials, predominantly in the form of antibiotics, are commonly and necessarily administered to production animals to just maintain the health of animal herds. However, that antibiotic use can also create an opportunity for bacteria in animal digestive systems to develop resistance. And as a result, bacteria possessing the genetic coding for antibiotic resistance, as well as any undigested antibiotics, are excreted in manure, with a common practice being using that manure as an organic fertilizer. And so when manure is applied to crop fields, the recipient soils and any downstream waters receiving runoff from these fields can create reservoirs for resistance where human exposure can occur. And so when we look at like kind of engineering a solution to this problem, sometimes it's really just easier to look broadly at these endpoint resistance reservoirs as just kind of a system of inputs and outputs. We've got the animal manure representing an environmental input, and what comes out downstream is the output. And overall, our goal is just to mitigate the amount of resistance contaminants that are at that output. And so with that goal in mind, we want to implement management practices that reduce the movement of resistance contaminants downstream. So practices that really work to keep the manure and its resistance contaminants in or near the place where they're applied. And then ideally, if that management practice can also help degrade any intercepted contaminants, that's an even bigger plus. And so it's here that my dissertation work actually kind of began. And essentially, we wanted to investigate the use of prairie strips as one of these potential management practices to reduce the transport of antibiotic resistance contaminants off of manured fields. And so I don't want to go too in depth here about what prairie strips are, because I think there have actually been other Iowa Learning Farm webinars with prairie strips as kind of the like main focal point. But just on the off chance that someone here has never heard of prairie strips before, they are a designated practice within the Conservation Reserve Program, whose agroecosystem benefits have been researched for over a decade. And prairie strips are essentially a unique form of vegetative buffer that incorporate diverse native prairie plant species. They're a cost-effective practice that can reduce water runoff, soil loss, and nutrient runoff, while also increasing plant, insect, and wildlife biodiversity. And so overall, mechanistically, prairie strips are able to increase the infiltration of runoff water and bolster the water holding capacity of soils by reducing flow velocity, promoting ponding, and increasing soil porosity. And so it was kind of through these already identified mechanisms and benefits that prairie strips were then also proposed as a management practice that could reduce the transport of AMR contaminants. So to kind of wrap up all of this background information that I'm giving to you, and before we get into discussing the research projects that I promised we'd talk about, I also just wanted to quickly go over the kinds of targets that we use for detecting AMR in the environment. And so these targets can kind of typically be separated into three categories that each kind of have their own pros and cons. And so the first type is just the antibiotics themselves. And these antibiotics can be what we call either medically important or non-medically important, with that distinction being based on if they are used also in human medicine. So some examples of medically important antibiotics, which maybe some of you have heard of because they are also used in human medicine, those include drug classes like penicillins. So those have active ingredients like penicillin or amoxicillin, tetracyclines, which include active ingredients like tetracycline or oxytetracycline. And then another would be like aminoglycosides, which has an active ingredient like gentamicin. And then maybe an example of a non-medically important antibiotic could be a class like the ionophores, which include the active ingredient monensin, which is a drug that is not approved for use in humans. And so while these antibiotics don't technically represent a direct detection of resistance in kind of the same way that the bacteria and genes we'll talk about do, we do know that the exposure of environmental bacteria to even low levels of antibiotics can result in a selection for resistance. And so detection of antibiotics can kind of be like the detection of a potential evolutionary driver of resistance, ultimately meaning that it's still an informative target. So the next type of contaminant that we might test for are antibiotic-resistant bacteria. And these bacteria that we isolate from environmental samples are essentially able to grow in the presence of a clinically relevant level of an antibiotic. And they represent what we call a phenotypic representation of resistance, because the resistance is an observable characteristic. The bacteria not only have the genetic coding for the resistance, but that genetic coding is being actively expressed, and the bacteria are visibly demonstrating resistance to an antibiotic. However, the issue with solely testing resistant bacteria as your target for AMR contamination is that when testing for these bacteria, you're looking for a specific target that you have to be able to culture and grow in the lab. And currently, environmental microbiologists estimate that less than 2% of environmental bacteria can be cultured in the lab. So our resistant bacteria targets are likely massively underestimating the total genetic presence of resistance existing in a sample's entire microbial community. And then finally, the last contaminant that I'll talk about are resistance genes. And so these refer to the specific pieces of DNA that code for antibiotic resistance. And they represent what we call genotypic resistance, meaning that there's a genetic potential for antibiotic resistance, but we don't necessarily know if it's being physically expressed, since we're not actually observing bacterial survival or growth in the same way that we are with resistant bacteria. And so the benefit of targeting resistance genes over the resistant bacteria is that we can kind of measure the total genetic potential of an antibiotic resistance in an environmental sample. But then obviously the downside is that we don't have a great way of differentiating between if these genes are physically being expressed, because they could be existing in a bacterial cell, they could be existing outside a bacterial cell. They could be present in a living bacteria, or they could be present in a dead bacteria. And even if they are present within a living bacteria, we don't actually know if they're turned on and being expressed, or if they're turned off and just kind of hanging out. So if it goes to the next slide, there we go. So thanks for bearing with me through that last slide. It's a lot of information, but I think it's really helpful moving into what I think is maybe the more fun part of this talk, and that's talking about our research projects. And so I'll preface that just for the sake of time, I'm going to be breezing through a lot of the methodology for these projects, but kind of my intent is to leave you with some of the bigger take-home messages. And so with that being said, I'll get to the first project, which we refer to as the flume experiments. And so a little background, a flume is an engineered structure where you can essentially control and measure the flow of water. And so here you can see a schematic of the real life flume that is kept in the basement of our building on ISU's campus. And so in addition to being able to, again, control that flow rate, you can see that our flume has this strips test section kind of right in the middle of it. And essentially through the work of another team member, Dr. Andy Craig, for our experiments, we were able to extract pieces from an already established infield prairie strip and then place those pieces into this test section of the flume. And kind of ultimately what we were trying to achieve with this setup was a modeled system in which we could control run-on flow rates and could produce runoff that had passed through a section of prairie strip. And so for some reference of what that looked like in real life, in this first picture, you can see our lab manager, Leanne, looking into the flume at an extracted and installed section of prairie strip with water running over the top of it. And from this, you can also see that our extracted pieces of prairie strip are relatively small. For dimensions, they were about 4 feet long, 3 feet wide, and only about 4 inches deep. And then in this second photo, you can see what's called our injection manifold. And this was used to essentially inject a dilute swine manure mixture into our flow upstream of the prairie strip. And it provided a source of contaminants that we could track with water movement through the prairie strip. And so using this flume system, we conducted experiments to test just how much prairie strips could reduce the concentrations of AMR contaminants under 3 different run-on flow rates. And overall, our hypothesis was just that the concentration of AMR contaminants downstream of the prairie strip would be reduced from the concentrations observed upstream of the prairie strip, but that the level of those reductions would be influenced by the run-on flow rate. And so for each experiment, a new section of prairie strip was extracted from a previously established strip site. It was transported back to our hydrology lab and installed into the flume. And then we got locally sourced swine manure and made a dilute manure mixture that consisted of roughly 1 part swine manure slurry to 40 parts water. And then this dilute manure mixture was added upstream of our prairie strip using that injection manifold that was pictured on the previous slide. And then over the course of each experiment, samples were collected upstream, downstream, and then beneath the prairie strip insert to essentially represent run-on, runoff, and infiltrated samples respectively. And so in order to quantify the kind of contaminant reductions that were caused by the prairie strip, we compared the concentrations downstream, so those at runoff and infiltration, to the contaminant concentrations that were seen upstream, so in the run-on samples. And then any change in concentration was labeled an effect of the prairie strip. And so during the flume experiments, we tested the various samples that we collected for antibiotic targets, resistant bacteria targets, and resistance gene targets. And what we saw was essentially a significant mass load reduction for all three. Essentially, retention of these contaminants by the prairie strip and removal from those surface runoff waters. And this result was certainly kind of what we had hoped for and kind of what we had expected to see. But it maybe wasn't all that exciting because it was kind of what we had expected. And so the more exciting piece that I think is that these three experiments also produced essentially three data sets that we could use to develop a mathematical model where you take a known run-on flow rate, a known incoming contaminant concentration, and then you can essentially calculate the necessary width of a prairie strip that would be required to achieve a certain level of contaminant reduction at that runoff. And so to kind of show you better what that would look like, this is a figure that was created by Dr. Craig that essentially shows a variety of scenarios and what length of prairie strip is required in each scenario to create enough contaminant reduction that the bacteria in outgoing runoff waters would be below the acceptable E. coli concentrations designated for Class A waters. So again, this is using some of that bacteria data that we generated. And kind of walking through this figure along the x-axis, you can see a range of incoming bacterial concentrations that would be moving off manured fields. The y-axis here represents the prairie strip width necessary to achieve the desired bacteria reduction. And then these different colored lines represent different run-on flow rates, with the orange being the fastest flow rate and the purple being the slowest flow rate. And so kind of a quick example of maybe reading a point on this graph, if you were looking at our orange line for a bacteria run-on concentration of 2,500 CFU per 100 mil, you would need a 20-meter wide prairie strip to achieve enough bacterial reduction so that your runoff water would meet the requirements of recreational freshwaters. So now turning our attention to these dashed black lines along our strip width axis, these represent the current design recommendations for prairie strips, which range from 30 to 120 feet. And so this figure represents what I think is maybe, again, kind of the more exciting piece to come out of the flume experiments. Because not only are we in the process of developing a tool that can help to maybe inform prairie strip design, but we can also see that of all of these modeled examples, including some really extreme events with really swift run-on flows and really high incoming bacteria concentrations, all of these scenarios are still falling within the already recommended width for strips. Meaning that in many cases, the currently established design width for prairie strips may already be effective at removing contaminants at a scale that makes the runoff water suitable for incorporation into recreational waters. And so I think that's pretty neat. We had kind of some expected results and then some really exciting conclusions from those flume experiments. But what we really wanted to do next was take our experiment out to the field and see if what we observed in kind of a modeled control system was also observed in an actual field setting. And so with that, we performed a rainfall simulation. And again, the goal of this study was to move from the flume scale to an in-field representation of prairie strips and to evaluate, again, the ability of prairie strips to just reduce the transport of AMR contaminants. And so to do this, we simulated rainfall events and created artificial runoff on field plots with swine manure amendment and prairie strip installments as our treatment factors. And so there were 3 treatments that we evaluated, shown here. And these included a section of non-manured crop with a prairie strip installment, which is referred to as strip plus no manure, a manured crop section with a prairie strip installment referred to as strip plus manure, and then finally a manured section of crop with no prairie strip installment that was referred to as no strip plus manure. And then 3 replicates of each of these were made for a total of 9 plots. So to begin, swine manure slurry was applied to the crop portion of appropriate plots. And then we also took some subsamples of this manure. And then a field portable rainfall simulator was used to generate runoff events on the various treatment plots. And then once runoff was observed, we took grab samples over the following 30 minutes of runoff. Of all these samples, we extracted the DNA and then analyzed that DNA for just a bunch of resistance genes that were chosen based on previous research and then also the known antibiotics that had been administered on the farm where the manure was sourced from. And so the heat map shown here is representing the quantification of our various gene targets in both the manure subsamples and then those runoff water samples. And so along this top x-axis, you can see this farthest left column represents our manure. And then the runoff water is just next to it, and it's subset into our 3 treatments. And then within each treatment, along this bottom x-axis, you can see the 3 replicates. So like plot 1, 2, and 3 all belong to this first treatment. Then our 20 targeted genes are listed along this left y-axis. And on the right side, you can see our legend showing that yellow is indicating a high presence of the gene in the samples, while purple represents a low presence. Then a grayed box means that we didn't detect the gene target in any of the samples. And they're very small, but these numbers inside the boxes are a representation of the number of samples out of 6 replicates where we had detection. So when compared by treatment, all of the genes quantified within the runoff water followed the same trend, with the no strip plus manure containing the highest relative abundance, followed by the strip plus manure, and finally the strip plus no manure. And so in short, every gene within our suite was significantly reduced from the manured runoff water that had first passed through a prairie strip. And so kind of as was seen in the flume, this was again certainly our expected result. For the non-manured plot to contain very low background levels of our resistance gene targets, our manured plot without any intervention to contain the highest level of the resistance gene targets, and then hopefully that manured plot with a prairie strip as an intervention would fall somewhere in between. But again, what I think is really special about these results is that not only was there significant reduction, but in 2 of our 3 replicates, plot 5 and plot 6, there was actually an almost complete removal, or at least reduction to below our limit of detection for all of the genes that we tested for. And so just overall, again, the strips are doing just a really impressive job at retaining these resistance contaminants and cleaning out the runoff that is eventually leaving them. And so with these first 2 projects, we identified the ability of prairie strips to intercept manure-derived AMR contaminants. And if you remember, that hits kind of our first goal of keeping the resistance contaminants near where they are initially being applied. However, the fate of those contaminants, once they are captured by the prairie strip, is another aspect entirely. And again, if you remember, ideally, what we want the management practice that we're using to do is not only keep the contaminants from moving, but we also want it to be able to degrade the contaminants that it intercepts. And so that brings me to the final project that we'll touch on, our soil incubation experiment, in which we wanted to investigate the risk of AMR contaminant accumulation within prairie strip soils. And so just to briefly describe the experiment, at 3 different strip locations, soil cores were taken from the area in crop and the area in strip. All of the soil cores from the same site and the same soil type were mixed together, and then 100-gram portions of each mixture were stored in mason jars. And then of all of these jars, half received a treatment of manure spiked with antibiotics, while the other half received a controlled water treatment. And these jars were then stored in control chambers and then were sampled at 6 time points over the course of the next 72 days. And similar to the rainfall simulation, our targeted AMR contaminant was a set of resistance genes that were likely to be introduced during the treatment of manure application. And so in order to kind of assess the rates of dissipation of these genes, we calculated what were essentially the equivalent of half-lives for 5 of the 6 gene targets. And while we didn't really see a distinction between the half-lives of the genes in the crop soils and the half-lives of the genes in the prairie strip soils, what we did see was that in both soil types, our estimated half-lives ranged from as short as 5 days to up to 10 days. And so what that means is that with the relative quickness displayed by these half-lives, we can kind of conclude that the turnover of introduced genes in the prairie strip soils is relatively fast, especially when compared to the frequency with which manure is applied to crop fields. And so that means that there may not be a large concern for the accumulation of these genes in the prairie strip soils over time. And so now that I took you really quickly through the various projects that we've been working on, I wanted to try and just recap what I thought are the important implications. So going back to the plume experiments, we learned that not only are the prairie strips effective at removing AMR contaminants from modeled run-ons, but in fact, the current design width recommendations for prairie strips may be effective at removing contaminants from this run-on even under extreme conditions. And then with the rainfall simulation, prairie strips were again shown to be extremely effective at removing AMR contaminants from run-on flows, but this time in kind of an actual field setting. And then finally, from our soil incubation, we saw that as prairie strips are removing AMR contaminants from run-on flows, it looks as if the intercepting prairie strip soils are also fairly effective at clearing those contaminants, and so their risk for accumulation may actually be quite low. And so just all in all, our research keeps showing that prairie strips do seem to be a well-suited management practice for reducing the transport of AMR contaminants within agroecosystems. And so lastly, before we kind of move on to questions, I'd like to just quickly acknowledge my collaborators on these projects, specifically Andy Craig, Jared Flater, and Alyssa Iverson. And then as well, I'd like to give a big thanks to all the members in both the Germs Lab and the Water Quality Research Lab. I also wanted to acknowledge the USDA NIFA grant that made all of this research possible. And with that, I'll wrap up. And while you're all hopefully typing some questions in the chat, here's the information for the certified crop advisors who are watching for credit. I'll come back to this slide in just a second so you can get everything copied down. But before I forget, I also wanted to plug the presenter for next week, Prashant Jha, who is a professor and extension weed specialist here at Iowa State, and looks like we'll be talking about cereal cover crop. So I'll go back to, hopefully, this slide. Yep, this seems like the important one. And then, yeah, any questions?
Right. Thank you, Dr. Alt. You did all of the work for me too there. You went through everything. So that's awesome. Okay. We do have some questions. So I'll try we'll try to get through as many as we can. The first one was, how might you expect your results to change in sandy soil conditions where maybe that organic matter and biological activity might be different?
Yeah, so I will say with that JAR study, the soil incubation study, I'll go back here, yeah. Something that we were actually looking into was the difference of kind of rates of dissipation between the crop and prairie strip. And I kind of just really quickly mentioned that we didn't observe a difference, but that wasn't really what we expected. So we definitely expect that different different soils will demonstrate different kind of microbial communities and different microbial activity. And our thought was specifically that the soils associated with prairie strips were going to be a lot more microbially diverse and then more resistant and resilient than kind of the crop soils that just have like this monoculture growth. But we didn't really see that, which was kind of surprising. And so I'd say that my assumption with like sandier soils is certainly that like mechanistically things might move quicker and kind of like deeper into the soils. But honestly, I don't know if there would be a big difference in kind of accumulation or not. My expectation is that there would be, but again, in the kind of soil incubation that we did, we didn't really see one. It may be something that we have to kind of take out to the field and really like keep those communities intact and give them the most environmentally relevant kind of chance at showing us a difference. But yeah, so I can't, I guess, say for sure how it would change, but I'd be really interested to see. I think you're muted.
Yes, I was. I was muted. Yes, if you can go back to slide 19, there's a question, I think a couple that kind of relate to that.
Maybe.
Well,
There we
19,
go.
right there, I think. Okay. It was, okay. It was about how wide was the prairie strip, and any idea why plot 4 looks so much different than plot 5 and 6?
Yeah. So the first part, width, I will say, going back to this slide before, maybe if I can. So these were essentially like 1 meter by 1 meter. And I will preface that like a comment that I've gotten a lot is that are these really representative of prairie strips, because it's essentially a like 50-50 ratio, which we don't really do. And I'll say that if we could do this all again, our intention was to do truly like a full field test of prairie strips where we funnel into an H flume and truly get like an actual infield representation. But we ran into some issues with like the person we were working with, their strip got like mowed and sprayed with herbicide and a bunch of different things happened. So we kind of had to really pare down to this. So I will preface with the caveat that these are strips that are essentially a 1-to-1 crop to prairie ratio. But why plot 4 was so different was that I don't know if there was a preferential flow path. We didn't really observe one, but it did have like a 10-fold higher flow coming off of it than plot 5 or 6 did. It had by far and away of all the 9 plots, the highest flow rate coming off of it. And so we're not entirely sure why that was. It didn't visibly look any crazy different than the other ones. But that definitely was likely the contributor to it being so dramatically different from plot 5 and 6.
Next one is, do you have results for the infiltrated water in the flume? Is there a groundwater or tile flow? Is groundwater or tile flow a relevant pathway for AMR?
Yeah, so if I can get back to the flume. So we do have data for the infiltrated samples. But again, with this, we were only, the size of the strip was only about 4 inches. So we didn't see a lot of actual like filtration or retention in the soils as much as we did kind of with that surface flow. But I will say with the rainfall simulations, we did also take soil samples. I didn't touch on this here because just for time's sake. But after we did the rainfall simulation, we went through and took soil cores from all of the plots and kind of separated it into 2 depths. So one was a 0 to 5 centimeter depth, and the other was like a 5 to 15 centimeter depth. And what we saw was that in the prairie strip soils, we actually had a higher AMR contaminant kind of concentration in those lower soils than we did in the upper. And then the reverse was true for the crops. So what we think is that with the prairie strips, it's kind of intuitive. The prairie strips have those like deep root systems that create a lot of like macropores and kind of increase infiltration. We think in the prairie strips, there's kind of deeper groundwater movement. which could be kind of a twofold thing. One, it could be, oh, great, we're doing a lot of filtration and that's a benefit, or it could be, oh no, we're just shunting this straight to groundwater. And so to kind of investigate that further, we're actually ongoing doing an experiment where we took like really big soil cores, like about that big, to a depth of 30 centimeters and then 60 centimeters. And we took those back to our lab and we essentially did kind of another sort of rainfall simulation and simulated like manure application rainfall simulation. And we're looking kind of more at how specifically the contaminants move through macropores in the soil, so we can kind of maybe resolve that system. And then with tile flow, that's another thing that we've looked at. So one of my colleagues, Dr. Liz Reiki, looked at tile flow 5 years ago, maybe now. And I think she did see that similarly, it can shunt those AMR contaminants into tile flow. And so that's another source that we have to be aware of.
Right. We're getting some great questions here. We'll try to get through a few more. One here is that a two-part question, I guess. Can the soil bacteria become resistant to antibiotics? And is there any method to eliminate the antibiotics from the manure? For example, a digestion system, composting, or manure sewage treatment systems?
Yeah, so that first part, can soil bacteria become resistant? Absolutely. Okay. And so I really briefly said just like this verbiage of resistance reservoirs. So bacteria actually do this kind of, I think, really neat, but also kind of insane thing where they can pass genetic information, not just parent to offspring, so vertically, the same way we do. They can also pass genetic information horizontally, meaning that if a bacteria comes into a close proximity with another bacteria, they have a potential through certain genetic mechanisms to share those genes. And so it becomes not just this like linear progression of resistance. There's also this almost, seemingly immediate progression where, again, just through proximity, bacteria can share genetic information. And so that's why it's like really important that we're wary of these reservoirs of resistance contaminants as well, because not all soil bacteria are human pathogens by any means, but just kind of the spread through a community is a really relevant danger. And then kind of it's just another driver, and it's another persistence of resistance where eventually it could come into contact with a human pathogen, and then that's bad news. And then the second part was what again?
Is there any method to eliminate the antibiotics from the manure? And somebody also, oh yeah, okay, go ahead. Yeah.
So the antibiotics themselves, I'm not so familiar with, but I know we are looking into, anaerobic digestion, composting, and then also like liming is a big one, heat treatment, solid liquid separation. We're kind of looking into all of those as methods for essentially killing off resistant bacteria and limiting resistance genes. I'll admit that I'm by no means a chemist, and so the antibiotics I don't touch so much, but I'm sure that some of those would have an effect. What effect that is, I can't quite say.
Great. Thanks. Okay. The next one is there in Minnesota, they're seeing a significant amount of runoff and dissolved contaminants during their frozen soil conditions, February and March. Your thoughts about prairie strip effectiveness for removing AMR during this particular period?
Yeah, so I'll preface that most of it is like speculative. But one thing we did do, so I'll start with this anecdote and then I'll kind of build off of it. But so with, again, the rainfall simulation, something we did do was it took place in late October. And so like I said, we went out and we got these soil samples. And our intention was to kind of continually go out and get like weekly soil samples, but the soil ended up freezing after like 2 weeks. So we got like 2 samples and then we had to wait like, 150 days or whatever until we could get the next soil sample because the soil was frozen. And so what we saw was that like by the time we went back out and were getting those thawed soil samples, the strips had done like a really good job of catching things, keeping things, and then, after all of that time, any of the manure specific bacteria we were looking for, I think had kind of died off in that freeze thaw. And so we found, almost negligible presence of manure bacteria after that freeze thaw. So it wasn't necessarily concern of, oh, it's trapped it, it's frozen, now it's unfrozen and snow melt is taking it with it. That didn't really seem to be the case. The other piece is that, again, I think it would be really interesting to see kind of what microbial activity we have going on in kind of different seasonalities, because certainly bacteria are likely to be more dormant or even dead in kind of the like frozen winter time when compared to like the prairies in full bloom, senescence versus growth kind of stuff. So from like a soil microbial community standpoint, I'd be really interested to see what's happening there. But from just a purely like mechanistic point of view, I mean, the prairie strips certainly senesce, but there is still that like vegetative cover that I think is really effective at keeping things in place and just slowing any runoff. And so I would hypothesize that they would still be pretty effective at least doing those mechanistic things of slowing down runoff and kind of increasing infiltration. But yeah, a lot of that's just kind of speculative. I'd be interested as well into looking into that.
Okay, great. And maybe close to the last one, is there concern for the contaminants being concentrated in the plant matter of the prairie strips that might be harvested for use in processing for feed or anything like that?
Yeah, so I'll say again with that column experiment that I told you guys about where we got the 30 and 60 centimeter columns. So there's a piece of that is looking at the soils and how things are transporting through the soils. And then another student, Grace Carey, is actually looking at that question specifically. She's looking at, is there any kind of association with the rhizosphere? Is there any accumulation in the roots, in the plants themselves? And so we are asking that question and we are currently looking into it.
Okay. That's great. And okay. And then there was one here. Did you test the manure slurry for AMR or get history from the farm? Which of the manure was used? What antibiotics were used?
Yeah. So for all of these, actually, we used the same local source, and we had just kind of a personal communication with the owner who could tell us what he was currently administering on the farm. And so we had antibiotic history, just like verbally communicated. We also sent those samples off to be analyzed for antibiotics, and they usually matched up pretty well with what he was telling us. There might be like kind of residual metabolites that we would detect, but then the knowledge of that would help drive what are we looking for. So like if tetracycline is being administered, we would maybe specifically look for tetracycline resistant enterococci or TET genes that confer resistance genetically to tetracycline.
Great. I think that in the interest of time, we will let Dr. Alt go there. I'll send you the chat. There were a couple of people that wanted to maybe contact you directly. So thank you, Laura, for a really important presentation. Thank you so much for boiling something that seems very complex to me into a very understandable, informative webinar. So thank you so much. With that, thank you everybody for attending the webinar today, and hope you'll consider tuning in again next week. And with that, Laura, any last comments?
Nah, just thanks everyone for listening to me go on and on.
All right. I'm going to